Welcome to our video on genetic mutations in disease. In this video, we will be covering the types and causes of mutations as well as two case studies, Tay-Sachs disease and sickle cell disease. Mutations are a change in the nucleotide sequence of an organism's DNA, which can have a range of effects depending on the protein that is ultimately produced. This is because the central dogma of molecular biology states that the DNA trinucleotide sequences determine the encoding of amino acids, which combine to express proteins. Point mutations occur when one nucleotide is changed into another. A no-effect point mutation occurs when the amino acid does not change, having no effect on the resultant protein. For example, if the GAA codon is changed to GAG, the translated amino acid is still glutamine. A missense point mutation occurs when changing one nucleotide alters the translated amino acid code. For example, GAG to GGG changes the amino acid from glutamine to glycine. Finally, nonsense mutation results in a stop codon. For example, GAA to TAA, causing termination of the reading sequence. Frameshift mutations alter the trinucleotide reading frames, which encode the amino acids, and as a result, the protein produced is usually non-functional. For example, if a uracil nucleotide is inserted here, the sequence is pushed along one nucleotide, altering the amino acids translated. This is called an insertion. Similarly, deletions and duplications have a chain reaction effect on the trinucleotide sequence frames, ultimately changing the protein produced. There are two main causes of mutations: spontaneous damage to DNA and exogenous damage from carcinogens. Spontaneous damage to DNA can be oxidative or hydrolytic. Oxidative damage occurs when the reactive oxygen species, such as superoxide radicals, cause damage to DNA by modifying the structure of the bases, inducing cross-linking or strand breaks. Hydrolytic DNA damage causes deamination and results from biochemical reactions of metabolites. On the contrary, exogenous damage to DNA is derived from external factors including chemicals and radiation. Free radicals are atoms with one unpaired electron and can contribute to the chemical damage of DNA. As they are reactive, they may cause breakages between the sugar phosphate backbone and the nucleotides of DNA, resulting in damage such as a double-strand break. Other carcinogenic chemicals which induce mutations include soot, cigarette smoke, and asbestos. Radiation such as ionizing radiation can directly affect DNA by breaking the DNA's glycosidic bonds. It can also ionize the water molecules within cells to create free radicals. Moreover, ultraviolet radiation can cause deformities in the DNA strand by forming dimers, which are covalent bonds between adjacent thymine bases. Furthermore, mutations may also arise from errors in nucleotide incorporation during DNA replication.
Tay-Sachs disease is an autosomal recessive disorder. The mutation for Tay-Sachs occurs in hexa, the gene encoding the alpha subunit of beta-hexosaminidase A, an enzyme. Beta-hexosaminidase A breaks down a fatty acid derivative called GM2 ganglioside. If hexa is mutated, beta-hexosaminidase A loses its functionality and GM2 ganglioside builds up in nerve cells, causing nerve death. 78 mutations in the hexa gene have been described, including single-base substitutions, deletions, and insertions. Since these mutations cripple the catalytic activity of hexa variously, there are several forms of Tay-Sachs disease, including acute, infantile, juvenile, and adult onset. Symptoms vary with the forms, but generally include motor function loss, mental disabilities, seizures, hearing and vision loss, and paralysis. Although there is no cure for the disease, some preventative measures, including genetic tests for partners, have been advised in high-risk populations. These ensure the mutated gene is not passed on to offspring. Sickle cell disease is an autosomal recessive disease caused by a missense mutation occurring in the oxygen-transporting protein hemoglobin. The mutation occurs in the 6th codon of the beta-globin gene, where adenine replaces thymine. Resultantly, the amino acid valine is incorporated instead of glutamic acid. The mutation causes a change in hemoglobin structure, resulting in the distortion of red blood cells into a sickle shape. These red blood cells die prematurely, leading to anemia. Sickle cell disease symptoms can also include pain in the chest, abdomen, joints, and bones. The inflexibility of sickle-shaped red blood cells also causes them to clog in small blood vessels, leading to painful swelling in hands and feet as blood flow to these parts is inhibited. Moreover, patients experience frequent infections as sickle cells can damage the spleen. Currently, bone marrow transplantation is the only cure. This involves drawing bone marrow from a healthy, genetically compatible sibling donor and transplanting it into the sickle cell disease patient. Thank you for watching our video on mutations and disease. I suggested the topic mutations and disease because I believe it is very relevant in contemporary society where disease is blatantly prevalent. My role in the team involved breaking down this topic into 4 sections and providing a brief outline of each section to ensure the information flowed logically. I also assisted in delegating specific sections to each team member and scheduled deadlines and meetings. My role also involved editing each team member's information and ensuring the overall information of the video was concise and cohesive by creating a script. I think researching the topic went well because there were many reputable sources available to us. The topic choice also worked well as we were able to assist one another if required, as we all understood the fundamentals of the topic. Moreover, the actual creation of the video works well because I found the program was easy to use and very aesthetically pleasing and professional. Perhaps one of the major challenges our team faced was online communications, as it was difficult to get everyone online simultaneously, and so communication lagged in this respect.
When we were first given this assignment, we really struggled to decide on a topic. We ended up going with genetic mutations and disease, as it allowed the potential to incorporate case studies, which is a great way of engaging the audience. It was also easily divided into categories for each team member, such as causes, effects, and the case studies. The next step was finding an engaging way of presenting the information. Initially, we were going to do a skit between a doctor and a patient, but we thought this would waste time on theatrics and wouldn't be as informative. We then found Powtoon, and although there were many technical difficulties associated with this program, it was a creative way of presenting our information. Although we learnt about mutations in BABS2204, I thought it was really interesting to link the mechanisms of mutations with the specific case studies. And finally, if I was to suggest anything to myself for my next group assignment, I would focus on assigning group roles, as this creates a more efficient way of tackling the project, and it would also give each team member drive and purpose in their part.
My name's Georgia. My role in the group was research for Tay-Sachs disease and the slides. The thing I found most challenging was probably trying to work out initially what we wanted to do and how to meet up with all of our different timetables and work and stuff clashing, as well as everybody having different life schedules. So sometimes I felt like a lot of the time that everyone was working and I was the only one that was out, so I wasn't always contributing. What worked well was then using Powtoon and Google Docs, because it meant that we could do it individually from home without having to meet up. And I think the reason we chose our topic was because it meant we could get a broad understanding of genetics and how conditions worked, and then focus that into 2 diseases. And yeah, I found it really interesting.
I have always found the concept of mutations to be quite interesting, so I wanted to find out a little bit more about them. It was quite easy to find a variety of information regarding this topic, allowing us to produce a useful educational video. Understanding mutations is a very important aspect of genetics and molecular biology, and since I'm interested in these areas, I took this opportunity to learn more about where my studies could take me. I was responsible for researching the causes of mutations and contributed to editing the script and video. Aside from some issues at the start when deciding on a topic and video concept, I found this project to be a relatively positive experience. As a group, we learnt how to overcome obstacles such as differing schedules and availabilities by using online collaborative tools such as Google Docs and Powtoon, which allowed us to contribute to the assignment in our own time to produce a high quality video.